I'm Daniel Levine, and this is the Bio Report. ProFound Therapeutics has developed platform technology to catalog and understand previously undetected human proteins and the role they play in health and disease. By focusing on proteins in the process of translation, the company said it's identified tens of thousands of undiscovered proteins that can provide untapped therapeutic targets. We spoke to Avak Kahvejian, co-founder and CEO of ProFound Therapeutics, about the company's platform technology, the vastness of the unexplored proteome it seeks to discover, and how this can lead to the development of breakthrough medicines. Avak, thanks for joining us.
Thank you for having me.
We're going to talk about ProFound Therapeutics, its effort to discover thousands of novel proteins and how this may lead to the development of novel medicines. How large a universe of human proteins is there, and how well mapped or understood is that universe?
Well, at the turn of the century, as you know, and many of your listeners might know, the Human Genome Project completed, and the whole purpose of it was to catalog every single base in the human genome, and to also finally come up with a complete list of the protein coding genes in the genome. And at the time, everyone was speculating that there was going to be maybe 100,000 genes, or some were saying 20,000 genes. We had some ideas from other organisms, namely things like the mouse and some other species. And so people were guessing in those ranges and hoping, of course, that we'd have many more than a simple rodent. It turned out after the completion that the team, the consortium, reported that there were about 20 some odd thousand genes. And that was kind of really bewildering to the community. But since then, the science didn't really advance much further, other than to identify maybe variants of those 20,000 protein coding genes. So the number now currently stands at something like around 100,000, but those are mostly essentially variants coming from those 20,000 original sites in the genome that code for proteins. So that was the current state of the art when we embarked on what became the science behind ProFound.
This reflects an emerging understanding of the vastness of the proteome. Why did people initially think it was much smaller, and how did people come to recognize its true breadth?
Well, the problem lay in how a gene was defined. So when the human genome completed, imagine what you're looking at are strings of A's, C's, G's, and T's. And you can imagine that there's no real interpretation of those. There are no landmarks to use to determine what a gene is and what a gene isn't. And so one had to come up with some rules to define the regions in the genome that coded for proteins. And some of the rules that people established were, well, we need a start codon and we need a stop codon. And so imagine those are the capital letters at the beginning of a sentence and the period at the end. And if we see those, then we can assume that there's probably a gene there. Those have to be 100 amino acids apart. They arbitrarily pick that cutoff. And we need to see that the gene is similar to a gene that we found in other organisms. So looking for what's called conservation. Those were really the three guiding rules that led to the annotation, the first annotation. And so that obviously was pretty logical way to proceed, because it gave you confidence that, well, if we've seen it in other animals and we see something similar in humans, it must be real. The capital letter and the period were important because the ribosome needs to see those things to put amino acids in order without stopping. And the 100 base pair cutoff, as I said, was somewhat arbitrary. But what we've realized since then is that those rules must have been quite constraining. And at Flagship, we asked the question, could it be that there are more proteins than meets the eye, and that maybe those canonical rules were a little bit too restrictive? And so if you lift some of those rules, for example, you say, well, what about things shorter than 100 amino acids? Surely there are proteins and peptides we actually know of today, that, such as insulin, for example, that are shorter than 100 amino acids. Why have we set that as the lower limit, or for defining a protein? And then other things like non-canonical start sites. If you will, there are other ways to start a protein and other ways to start protein translation that don't have to begin with the traditional AUG start code hub. If you expand to using different codons to start the sentence, you actually end up broadening the landscape even further. And so these were some of the things that we ended up doing, combined with novel technologies to actually read the active portion of the transcriptome that is being converted into protein. And through that, we were able to see that there were indeed tens of thousands of additional genes or protein coding RNAs in the genome. And that's what got us so excited about this opportunity and the expanded field.
You're focusing on proteins in the process of translation. Can you explain what you mean by that and how that might create new opportunities?
Well, as you know, in the central dogma, DNA is converted to RNA in the nucleus. That RNA is called messenger RNA because it leaves the nucleus and serves as the message or the template for another conversion step known as translation. And that's where the mRNA engages with what's known as the ribosome, and the ribosome reads the mRNA and uses the codons in the mRNA, 3 nucleotides at a time, to add amino acids back to back to create an amino acid chain, which becomes a protein. And that central dogma is fundamental to life. It's how all proteins get made in every kingdom of biology. So the normal way people have looked for proteins has either been at the protein end, looking for proteins in samples or in tissues or in serum, let's say, in circulation, or the other way, which we talked about, looking for the code in the genome. But there's a middle ground where you can actually trap the messenger RNA as it's being translated, as it's being converted into protein. And that arguably is one of the best ways to find proteins, because you don't need to find the protein itself in the first instance, nor do you need to create maybe arbitrary rules to find the blueprint for a protein in the genome. You're actually finding the mRNA and the ribosome in the act of converting to an amino acid sequence. So that's what we call translatomics, the science of identifying transcripts, mRNA transcripts or RNA transcripts that are getting converted into protein sequences in real time, catching them in the act, if you will.
What's the implication of this from a therapeutic development point of view?
Well, first, from a scientific point of view, it allows you to see more proteins than we've ever identified before. And proteins are the building blocks of life. Every single thing that our body does is done via a protein more often than not. The ribosome itself, the machine that's reading the RNA to make proteins, is made of protein and RNA. Enzymes that we use for a variety of biochemical functions are proteins themselves. every single actor in human biology is a protein. And more importantly, every single drug that we take either hits a protein and debilitates it, or is a protein itself. And so when you look at the world from that lens, proteins matter, and they matter a hell of a lot. And we've been limited to date to the 20,000 protein coding genes. The pharmaceutical industry, the biotechnology industry, has essentially focused its entire effort on a subset of the 20,000 protein coding genes in the body as drug targets or drugs themselves. And obviously, we've come up with amazing drugs thanks to that paradigm. But what if there were more than those 20,000? And that's really the question that ProFound is putting forth. If there are more than those original 20,000, that opens up the landscape for new disease targets, and it opens up the landscape for potentially new drugs, new proteins that can be bioactive in and of themselves, and we can make and take to cure disease.
ProFound was founded by Flagship Pioneering's Flagship Labs in 2020. It was unveiled in May. How did it come about, and at what point did Flagship see this as technology to build a company around?
Well, Flagship, as you mentioned, has a unit which we called Flagship Labs. We've been originating companies since the 2000s, early 2000s. And the way we do that is by undertaking what we call explorations. We're all scientists at Flagship, and we have a deep curiosity in human biology and the life sciences broadly, and identifying opportunities to build new technologies and bioplatforms that can have an impact on human health, on sustainability, etc. And we have been conducting explorations in a wide array of fields. That's led to a number of companies that have been originated completely in-house, where the intellectual property, the science, the founding pioneering vision has all stemmed from our collective effort to ideate, to posit, to make leaps of faith into the unknown, and then to reduce that to practice and move into an inventive step of creating that new technology. So ProFound was no different. We conducted an exploration into this whole notion of, is there a dark proteome, a collection of proteins we just cannot see with the current technologies at our disposal. And we started to look into things like mass spectrometry, which Flagship has a very long history with, a technology that's used to detect proteins directly. And we saw that mass spectrometry sees a bunch of stuff that we can't really parse. We looked at genomics, and we looked at as I mentioned, the Human Genome Project and how they applied these arbitrary rules to define a protein coding gene. And we also have a very long and deep history with mRNA translation, being the founders of Moderna and the company that essentially harnessed that step in the central dogma to create medicines inside the human body. So that's that kind of exploration and connecting the dots across the central dogma, and the notion that new technologies were available to look for proteins in an orthogonal way, independent of the limitations of mass spectrometry and other proteomic tools, was what spurred us on to actually venturing into not only hypothesizing that new proteins might exist, but actually going to hunt for them. And as we started to do our own analysis, and as we started to conduct our own experiments, it started to bear true. We started to see actually signals from biological samples telling us that these proteins, new proteins existed that no one had ever seen before. And that's what led to the formation of the company and became the impetus for us to create a broad bioplatform to pursue this in earnest.
Well, what is the ProFoundry platform, and how does it work?
So the ProFoundry platform includes a lot of the things I touched on. There is a component of omics technologies. One particular form is the translatomics platform, the ability to see RNA transcripts being converted into protein and catching them in the act of getting converted to protein. But that's complemented with mass spectrometry to identify the proteins themselves directly. It's complemented with computational tools and DNA sequencing tools, really to span the central dogma from DNA to RNA to protein and capture that entire biology. But what's more, once you've identified these proteins, you want to understand what they do. It's not enough to just see the pieces of the puzzle or to find the building blocks. You need to understand where they fit and what their role might be in disease and health. And that's where we have additional technologies and capabilities to, in parallel, query the functions of these things and perturb biological systems to understand how they may or may not play important physiological roles. That's going to be the key to figuring out whether they should be bona fide targets for drug discovery or whether they can be drugs in and of themselves. So that's part and parcel of the ProFoundry platform. So you can imagine a number of omics tools combined with computation and experimental systems that are at a throughput and parallelization that is required to tackle this landscape at scale.
It's one thing to identify a novel protein. How do you then tease out its function and its potential as a therapeutic target?
Well, if you have these building blocks, you now need the tools to perturb the building blocks. So imagine you remove one of these things from a biological system, or you add them back to a biological system. These are the kinds of things we're doing, and we're doing them at scale. So some of the tools at our disposal include things like CRISPR-Cas9, which can go and quote unquote surgically delete one of these genes from a cell. And we can then read out how that impacts the cell. Does the cell grow more quickly? Does the cell change its behavior and change its shape? Does the cell die? And all of those things inform us as to what is that particular protein doing. Now, if you did this one by one, it would take a long time. And so that's where scale and parallelization matter quite a bit. And as we go through these, what we call functional genomics efforts, we then move into a regime where we can start to perturb the systems with the proteins themselves, adding them back and then testing them in animals and so on and so forth. All of that is built in-house and helps us decode their function and how essential they are to a particular pathway, how disruptive they may be to another pathway, so on and so forth. And who are they interacting with? What are these building blocks engaging with inside the cell, inside the body? And those are all the tools we use to essentially identify the smoking gun, if you will, and decide whether or not to prioritize one protein over another for its involvement in disease. Now, we also use a lot of computational tools, and we use publicly available information. There's a lot of information about mutations in the genome and their relationship to disease. We can also come at it that way to figure out which of these particular sites in the genome that have heretofore been ignored, we can shed light on those and implicate them in disease thanks to this new information.
You've been able to already use the platform to develop a growing list of novel proteins. What have you been able to find, and have any of the novel proteins you've identified provided significant leads for potential therapies?
Well, using all the tools that I mentioned, we've identified novel enzymes, novel receptors, different kinds of immune modulators. All of these are essentially extensions of the various functions we've seen with the traditional canonical proteome. So that has us very excited that we've identified things that look and feel and act like the proteins that we've all grown comfortable with in the canonical proteome. Now, the trick is to, as you mentioned, figure out which ones we want to start to drug and which ones we may want to use as drugs. And so that's the phase we're in right now. We're we're seeing similarities. We're seeing that these things are that they have a kinship with the existing proteins, and that's helping us figure out their function. The next phase is figuring out which ones are actionable.
In the past, we featured Matthias Uhlin of the Human Protein Atlas on the show. There are others seeking to identify novel proteins as well. How competitive a landscape is there for what you're trying to do? And is there a race to identify and exploit these proteins?
Well, I think we've seen it as a sign of encouragement that the academic community has now gotten excited about this field as well. The ENCODE project, which is a large consortium of various laboratories throughout the world to help understand the genome and to help map the genome, has started to work on this and to publish on this as well. So yes, we're all going after this vast landscape, but we think the landscape is large enough that there's plenty of room for innovation and opportunity. And we're doing it in a way that's much more oriented towards disease, towards finding practical solutions in health, and applying a lot of tools at scale to do that. And so we're actually excited by the fact that this new field of science is gaining recognition and momentum.
There's such a large universe of potential proteins you're looking at. It strikes me that with all this richness, this challenge is really how to prioritize what you will pursue for therapeutic development. How do you go about doing that?
Well, part of it is to bring the brain trust of Flagship to bear on thinking about unmet needs in the disease landscape and in human health. The other is to think about model systems and which diseases are easy to model in a laboratory, which cell types are more readily perturbed in the laboratory setting and easier to study. So some of it has to do with having insights and mapping onto the broader opportunity landscape, and some of it has to do with practical considerations for making progress quickly. So taken together, that's partly how we've been deciding what to work on and what areas to prioritize over others.
What's the business model here? To what extent will you rely on partnering, and to what extent will you develop your own pipeline of therapies, and how far through development would you plan to take them?
Well, as you mentioned, this is a vast landscape. So we think there are going to be things that we do ourselves, of course. And when I say that, I mean take them all the way. We're pursuing things that we would want to develop drugs against, and we're going to be making some of these proteins into drugs themselves and taking them through the clinic and eventually to market. So we're going to be fully vertically integrated in that respect. But at the same time, there's so much opportunity and so many different diseases that one could look at through this lens that we think it's going to be ripe for doing multiple partnerships, either within the Flagship ecosystem or outside of the Flagship ecosystem. And we think that different entities, organizations with particular interests in a different set of disease areas will be complementary to the skill sets and technologies that we've brought to bear, so that we can tackle a broader swath of diseases and unmet needs through collaborations and partnerships.
You mentioned the Flagship ecosystem. Flagship has a large network of companies it's backed. What are you doing to leverage those relationships?
Well, as you can imagine, we're involved in origination across a wide array of bioplatforms, biological systems, and disease areas. So we are essentially exposing the community here to what ProFound is doing, and engaging in dialogue with all of the various startups that we have in our ecosystem. These startups are, some of them, wholly owned subsidiaries of Flagship, and the partners that are leading them or are involved with them are my colleagues, and we sit on the same floor. So there's a really tight connection among the various companies and scientific projects. And so there's an openness and a willingness to cross-collaborate, share information and insights, and even conduct projects together. We also have a group inside Flagship known as Pioneering Medicines. And Pioneering Medicines is essentially looking at at the broad swath of bioplatforms we have at Flagship and looking for opportunities to develop drug programs and assets out of these companies. And so we're working with Pioneering Medicines quite closely to identify early opportunities that we may want to accelerate through their platform.
Flagship has made an initial commitment of $75 million. How far will that take you, and how are you thinking about raising additional capital?
Well, as is always the case, Flagship is the sole financier, if you will, allocating resources to the company to prosecute the science, to build the bioplatform, and to build the original team. And then thereafter, we'll support the company through its various phases and stages. But we will invite others to come along on the journey with us. And we're going to expose other investors to the science, the data we've generated, the new proteins we've identified, and the opportunities that they promise, and essentially raise more money to prosecute those things towards the clinic. So that money in and of itself won't be the only monies we'll bring to bear on this. And so that's more to come in the coming year or so. That'll help us expand our efforts.
Avak Kahvejian, Flagship Pioneering General Partner and co-founder and CEO of ProFound Therapeutics. Avak, thanks so much for your time today.
Thank you. It was a pleasure.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send an email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.